Okay, welcome to yet another one of the senior capstone talks. We have 23 of them this term, and so far we've heard from 2 people, so we have lots more talks. And so I want to encourage you all to come. Anna and Jamie will be speaking in this very room starting at, Anna will be at 3:10 and Jamie at 3:45. You can go on to on this Friday. Thank you very much. I forgot to mention the Friday part. I'm still on California time. And if you check the front page of the Lawrence website, you can see them in the calendar and read about what read their title and things like that. And it's my distinct pleasure to introduce Mary. And in some ways, I feel I'm kind of substituting for Ron Peck. But to start out with, I wound up having Mary as an advisee freshman year. She walked into my office and we hit it off very nicely. And that have worked together over the past 4 years through lots of trials and tribulations, a variety of different things. Not one of which was losing Ron to Colby College. And that it still bugs me to this day that that all 3, Nick, Mary, and Emily, I had a full lab at that time. And so all 3 of these freshmen went to Ron's lab. And I was like, oh, if I only had more money. And such is life. Anyhow, Mary has found lots of excellent research opportunities to pursue. And so that she'll be telling you a little bit about her research at Mayo. She is planning on doing an MD PhD, but for now is going to do a little gap year because she loves research so much with Kim Dixon upstairs, who has money and I don't. Anyhow, without further ado, Mary.
All right, hi everyone. Today I'll be talking about the research I conducted at Mayo Clinic this past summer, and we were investigating the regulation of endosomal trafficking during tumorigenesis. So a little bit of background information for you guys. Here I've depicted a cell in blue. So white represents outside of the cell, blue represents inside of the cell. The orange and green shapes represent cell surface receptors, and cell surface receptors are really important for cells to carry out everyday functions that they need to survive. These little colored ovals I have up here binding to the cell surface receptors are called ligands. And ligands are bound to a receptor and then activate that receptor, which I've represented with the yellow shape. Once a receptor is activated, it can further send signals throughout a cell and activate other cellular processes that are in charge of many other functions for that cell. So it's important for cells to be able to regulate these cell surface receptors because they are in charge of so many other functions. So there are a few ways to regulate cell surface receptors. Here I've shown the orange receptor being enveloped into an endosome, and the same goes with the green receptor. And then there are 2 major pathways where they can be regulated. One, which is the green receptor being sent to the cell surface, called recycling, and the other I've shown with the orange receptor being sent to the lysosome to get degraded. However, this degradation pathway is not that simple. So if you see the orange receptor in the endosome, that tail is still exposed to the blue cytoplasm, and this allows that receptor to continue sending out signals and activating those other cellular processes. And this does not allow the receptor to be sent to the lysosome for degradation. So what we need is a type of intermediate vesicle that can contain the entire receptor, stop that signaling, and therefore transport the receptor to the lysosome. And this type of intermediate vesicle is called a multivesicular body, or MVB. And an MVB is just what it sounds like. So it's a specialized type of endosome, and it contains multiple interluminal vesicles. So what happens is the endosome sends the receptor to the MVB. Once the receptor is in the MVB, it contains the entire receptor. So you'll see that that tail is no longer exposed to the blue cytoplasm, and therefore there is no more signaling from that receptor, and then the receptor can be sent to the lysosome where it can get degraded. So a question you might have is, how does a multivesicular body know where to transport the receptor that it contains? And this is answered with escorts, a kind of fitting title for that purpose. Escort stands for endosomal sorting complexes required for transport, and there are 4 of them. They are involved in other cellular processes besides MVB sorting, such as autophagy, exosome formation, viral budding, and cytokinesis. And how an escort works is by recognizing a ubiquitin that gets bound to the receptor in the endosome, and then the escort can recognize the ubiquitin, bring that receptor into the MVB before sorting it into the lysosome for degradation. So this is a visual of what I just said on the previous slide. So looking at number 1, we have the orange receptor with a ubiquitin bound to it. The ubiquitin is recognized by the escorts in the MVB. The escorts bring that receptor inside of the MVB, and then the MVB can sort that receptor into the lysosome for degradation. Just a little bit of more information on those escorts. So like I said, there are 4 of them, and they're called 0, 1, 2, and 3. Escorts 0, 1, and 2 are preformed complexes that are recruited as a whole to that MVB membrane. And escort 3, which is the one I'm most interested in, is a dynamic polymer. So what this means is that it can change from a membrane-bound form to a soluble form. That's where the dynamic comes in. And polymer means that it has multiple subunits. So in this case, it has 7 subunits. So here I've zoomed in on the MVB, and I'm showing escorts 0, 1, and 2 lining up those orange receptors on the MVB membrane. And then escort 3, which is this purple spiral, is the component that actually brings those receptors inside of the MVB. So one of the important reasons why we study escorts are because they're involved in regulating many different cellular receptors, and therefore many cellular processes are also regulated indirectly from escorts. So if there is a mutation or depletion of an escort, it can cause diseases in the human body, such as cancer, neurodegenerative diseases, and even infectious diseases. So it's really important that we can understand the mechanisms that they work by. So like I said before, my project focuses mostly on escort 3, and here I've shown it on the membrane and then being converted into its soluble form. And the way that escort 3 gets converted is with the help of an enzyme called VPS4. So VPS4 is an ATPase enzyme, which means when it's functioning, it needs energy, which is ATP, and then it converts the ATP to ADP. This other protein up here called VTA1 is a cofactor to VPS4. So what a cofactor does is just assist that VPS4 enzyme while it's doing its job. So this diagram is representing the pretty complex relationship between VPS4, escort 3, and VTA1. And what I want you to take away is that VPS4 can interact directly with escort 3, or it can interact indirectly with the help of the cofactor, and that all of these components are necessary for the functioning of the other. So the approach of my project involved making mutations of VPS4, the ATPase enzyme, and also VTA1, the cofactor, and then analyzing what happened. So I was looking at, if we mutate VPS4, can it still function as an ATPase, converting that ATP to ADP? If we mutate VPS4, can it still interact with its cofactor VTA1? If we mutate VPS4, can it still disassemble escort 3 from the membrane? So these are the 3 different parts of my project. This top row is looking at mutating VPS4 and then seeing what happened compared to wild type. The second part of my project looked at the interaction between the mutant VPS4s with the cofactor VTA1, and also analyzing what happened with respect to its ATPase activity and the disassembly of escort 3. This third part of my project looked at mutating both VPS4 and the cofactor BTA1, and then seeing what happened with regards to the ATPase activity and the disassembly of SCOR3. So there were 3 different versions of VPS4 that we made, and they each were a different type of point mutation. A point mutation is when you change a single amino acid in the protein. So for example, up here, we mutated the methionine on the 330th position to lysine. The next one was L407K, which is the leucine on the 407th position to lysine. And then the final version of VPS4 that we made was mutating the lysine on the 411th position to alanine. So the reason we chose these residues to mutate were because they were suggested via modeling that a previous student at Mayo had discovered, and they occur right outside the domain on VPS4 that interacts with BTA1, that cofactor. These 3 point mutations are all non-conservative, which means we are changing the characteristics of the amino acid. So for example, in M330K, we are changing methionine, which is a hydrophobic amino acid, to lysine, which is an amino acid that has a positive charge on it. There is one different form of BTA1 that we made, and it contained all 3 point mutations. And as you can see, we changed them all to an E, and E stands for glutamic acid. Glutamic acid has a negative charge, so we are also making non-conservative mutations of the cofactor BTA1. These residues were also chosen because they were suggested via modeling, and they actually occur inside the area on BTA1 that stimulates VPS4. The first method we chose in my project to use were Atpase assays. So if you remember, I told you that VPS4 is an ATPase enzyme. So when it's functioning properly, it will convert ATP to ADP. So the first Atpase assay we did was comparing the mutant forms of VPS4 to the wild type form of VPS4 and seeing how efficiently it was able to convert ATP to ADP. The second Atpase assay that we did was looking at the interaction of the cofactor with the mutant forms of VPS4. So we wanted to see if we mutate VPS4, is the cofactor still able to interact? And if so, how is the effectiveness of VPS4 as an ATPase? The last ATPase assay that we did was looking at mutating both VPS4 and the cofactor BTA1. So if we mutate both of them, are they still able to interact? And if they are able to interact, what type of ATPase activity do we see? So when we were doing this ATPase assay, we were using radio-labeled ATP, and we would mix this radio-labeled ATP into our samples. Our samples would be VPS4 mutants alone, or the VPS4 mutants with the cofactor BTA1. Once the radio-labeled ATP was added, we would place our samples on a TLC plate at specific time intervals, and then we would let the samples migrate up the TLC plate for a set amount of time. Once this time was passed, we could visualize the plate to determine how much ADP was present, and then quantify the amount of ADP that was present because the ADP was radioactive, so we could visualize the brightness of that radioactivity on the plate. So this is the first graph of data that we got from the first Atpase assay. Y-axis is looking at the amount of ADP per VPS4 per minute. So this is really just showing how functional is VPS4 at its ATPase capability. The x-axis is looking at the concentration of VPS4 in micromolar. So if we look at the black line, that represents wild-type VPS4. So that shows us what a normal VPS4 should function at those varying concentrations. The red, blue, and green lines are showing us the 3 different mutant forms of VPS4 that we made. And what we wanted to show with this assay was that we didn't mutate a part of VPS4 that was necessary for its ATPase activity. And as you can see, that's what we saw with this graph. So the red, blue, and green lines are all very similar to that black line, the wild type. which was good. We didn't want to completely alter its enzyme activity. This is the second graph of the second ATPase that we ran. And in this case, we're looking at the fold-stimulated ATPase activity on the y-axis. So what we did to get this was take the VPS4 plus the cofactor and then subtracted the VPS4 alone condition from it. Because what we really wanted to look at was how much VPS4 stimulation is due solely to the cofactor. So looking at wild-type VPS4 interacting with wild-type VTA1, we see about a 3-fold increase in its ATPase activity. This is what we would expect because it's normal VPS4 and normal cofactor. When we look at the blue bar, which is the M330K version of VPS4, we see about the same stimulation as wild-type. And this would suggest that when we mutated VPS4 on the 330th position, we did not interfere with the cofactor's ability to bind, and we still see the same stimulation as wild-type. However, when we look at the L407K and K411A mutants, we see reduced stimulation compared to this wild-type. And this would suggest that when we mutated VPS4 on the 407th position and 411th position, we changed residues that were necessary for that cofactor to bind VPS4 and stimulate it. So this graph is very similar to the last graph I showed you. The only difference is that instead of wild-type VTA1, we're using that mutant form of VTA1. So on the y-axis, we have that fold stimulation again, and this will be the amount of stimulation that's due to the cofactor. Looking at the wild-type VPS4, we only see about a 1-fold increase in stimulation, and we would expect this because we mutated the cofactor in a way that would not allow it to interact normally with wild-type VPS4. And the same goes for the green bar, which is the K411A version of VPS4. It was not able to interact as well with the cofactor VTA1, and therefore we didn't see the same stimulation. When we look at the M330K and L407K versions of VPS4, we see hyperstimulation compared to wild type. So if you remember the VPS4 residues, we changed them to lysine, which has a positive charge. And the mutant form of the cofactor, we changed those all to glutamic acid, which has a negative charge. So what we think is going on here is that the positive charges on VPS4 can interact with the negative charges on VTA1 and create a compensatory mutation that allows for an even better bond between VPS4 and the cofactor, and that's why we see that hyperstimulation. The next part of my project involves subcellular fractionation, and this allowed us to look at whether escort3 was in its membrane-bound form or its soluble form. So in order to do this, we would remove the yeast cell wall and generate spheroplasts. And then the next step was lysing those spheroplasts. So lysing means we would pop them open, but we would do it very carefully so we wouldn't damage anything in those yeast cells. Next, we would separate the soluble escort3 from the membrane-bound escort3, and we would do this by spinning our samples. So when we would spin our samples, the supernatant that would form would be all of our soluble material, and the pellet that would form at the bottom of the tube would be all of our membrane-bound material. And from now on, I'll refer to P13 as the membrane-bound material and S13 as the soluble material. The next step involved doing a Western blot of all of our samples, and the Western blot allows us to look for a specific protein. In this case, we are looking for escort3. We would probe for SNF7, and SNF7 is just the most common subunit of escort3, so it's a really good marker for us to determine whether escort3 was in that soluble form or its membrane-bound form. So just a little recap. We have escort3 in the membrane-bound form here at the top, and VPS4 converts it into its soluble form where all of the components are separated. And to orient you guys to the Western blot, this top row is going to be looking at escort3, and then we'll have two columns. We'll have the S13, which is the soluble, and P13, which is the membrane-bound. And then this label up top is telling us what type of BPS4 I was using. So in this case, I was using wild-type BPS4. So if you look back up at the picture, when we have wild-type BPS4, we should be able to convert all of the membrane-bound escort3 into the soluble escort3. So if you look at the blot, we have a nice dark band in our soluble escort3 and very, very little left in that membrane-bound, which is what we would expect with a normal BPS4. It would be able to function normally and disassemble escort3 from the membrane. This next column is looking at a strain of yeast that was missing BPS4 completely. So if you look back up at this picture, if we're missing BPS4, we should not convert escort3 to the soluble form, because the only way it can be converted to its soluble form is with the help of that enzyme. So as you can see in the blot here, we have a really dark band under our membrane-bound form, which suggests that none of the escort3 was converted to its soluble form, which is also what we would expect because we are missing BPS4. This next column is looking at how well our complementation worked. So this was a strain of yeast that was missing BPS4, and then we added back in the wild-type version. So if we added in wild-type, we would expect to see the same conversion of membrane-bound escort3 into that soluble form. However, you'll see we did not see the same band pattern as we did in the actual wild-type version. We needed to run some more replicates of this to see what was going on, but we ran out of time and were unable to do so. These last three columns are looking at the mutant forms of BPS4. So if we mutated BPS4, we wanted to see how functional it was at converting the membrane-bound form escort3 into the soluble escort3. So what we see here is kind of a 50-50 split between that soluble form and the membrane-bound form. And this would suggest to us that there's an intermediate phenotype of BPS4 that we found with mutating those residues. This bottom row of the blot is a soluble control. So what we wanted to do was make sure our soluble samples were only soluble material and that our membrane-bound samples did not have any of the soluble membrane-bound or soluble escort3 in it. So what we did was probe for PGK1, which is a known soluble protein. And as you can see, it shows up only in our S13 samples, which is good, which means out of all of our samples, our soluble samples truly were soluble and our membrane-bound samples truly were membrane-bound. There's no mix. The last part of my project included microscopy, and this would allow us to see the functionality of escort3. So if you remember, escort3 is the component that brings those receptors inside of an MVB. So BPS4 is necessary for escort3 to carry out its job in bringing those receptors inside the MVB. So what we did first was take a strain of yeast that was missing BPS4, and then we added in our mutant BPS4s and GFP-CPS. So GFP stands for green fluorescent protein, and we tagged this protein to CPS. And CPS is just a type of receptor. So what we did was put green on this receptor so we could visualize where it was with respect to an MVB. Next, we labeled the yeast with FM464, and this allowed us to visualize the membrane of an MVB. Finally, we were able to take images of the cells using deconvolution microscopy, and we used a 100x objective lens. So these are the pictures that I got of my microscopy. This top left image is looking at wild-type BPS4. So if we have wild-type BPS4 around, it should enable escort3 to do its job, and that's why we see all of those green tag receptors on the inside of the MVB. So escort3 was able to do its job and bring those receptors inside. The top right picture is looking at a strain of yeast that was missing BPS4. So when BPS4 is missing, escort-3 can't do its job, receptors are not being brought inside of the MBB. And that's why we see a nice green outlining that MBB, which means all of the green tagged receptors are stuck on the outside and they're not being brought inside. This bottom row is looking at the 3 different versions of BPS4 that we made. So what we wanted to find out here was if we mutated BPS4, how functional was escort-3 at bringing those receptors inside. So you'll see that there's a pretty defined ring on all of these pictures. And this is similar to the strain of yeast that was missing BPS4 with that nice defined outside. And this suggests that some of those green tag receptors are stuck on the outside of the MBB. But you'll also notice that there is some green on the inside of these as well, which is characteristic of wild-type BPS4. So some of those green tag receptors were still able to get inside the MBB. And this is also characteristic of that 50-50 phenotype that we saw on the Western blot. So we've got some receptors on the outside that are stuck, but some of those receptors are able to get inside. So there are a few conclusions that we can draw from my project. Overall, we can say that the 3 residues we chose on BPS4 did not globally compromise its ATPase function. So if you remember that first graph, the mutants were still able to convert ATP to ADP at the same efficiency that wild type was. Next, we can say that the cofactor BTA1, the wild type version, was not able to enhance L407K or K411A. And this would suggest that when we mutated those residues, we interfered with the cofactor's ability to bind, and therefore we didn't see stimulation because we interfered with that bond. On the other hand, when we mutated M330K and L407K with those positive charges, and we had the mutant form of the cofactor with those negative charges, we created a compensatory mutation, allowing those positive charges and negative charges to interact, and then we created an even better bond between BPS4 and cofactor, which hyperstimulated its BPS4 ATPase activity. We can say that the BPS4 mutants that we made show an intermediate phenotype. Now, it's hard to quantify an intermediate phenotype, but if you remember back to the Western blot, we saw the 50-50 split between escort-3 being on the membrane and escort-3 being in its soluble form. And then if you remember back to the microscopy pictures I just showed you, we had some of those green tag receptors stuck on the outside of the MBB, but some of them were still able to get inside of the MBB. Overall, we can say that M330, L407, and K411 contribute to BPS4 stimulation via escort-3 and BTA1, which is a start to identifying which residues of BPS4 and BTA1 are responsible for stimulation and disassembly. Overall, it's important for us to understand the regulation of this endosomal trafficking because it could lead to the development of new targets to more efficiently treat some of those diseases that I was talking about earlier. There were a few other steps I would have liked to have taken with my project if I had more than just 10 weeks to work on it. If I was given more time, I would have liked to have made more mutants of BPS4 and BTA1. So our original goal was to identify the specific residues that are involved in the ATPase activity, the ability of escort-3 to be disassembled, and that interaction between BPS4 and BTA1. And then I would also like to have made more mutants of BTA1 to see just where that interaction between the cofactor and the enzyme is. I would have also liked to have used X-ray crystallography to determine structures of BPS4 and BTA1. So you probably noticed on the slides where I introduced those two to you, they're just shown as rectangles. And that's partially due to the fact that we don't know their definite structures. So I think that would have been a cool next step to take to try to identify these, how they interact better, the structures. I would like to thank my buddies over at Mayo Clinic, specifically David Katzman, who was my PI, and Brian Davies, who helped me out a lot in lab, and then also to Shirley Dean and Jason Tan, who are currently grad students over there right now. here at Lawrence, I'd like to thank Dr. Dave for being my advisor for the past 4 years and helping me with this project once I was back on campus, and for also introducing me to the world of virology, getting me started in his research. I'd like to thank Kim Dixon for getting me into her lab as well, working with angiogenin and ribonuclease inhibitor. I would like to thank Professor Peck, who took me under his wing as a freshman and really introduced me to the world of research. And finally, I would like to thank Emily Hoyleman and Nick Wallner for being the best research buddies over the past 4 years. Thank you. Questions? Yeah.
What are some receptors that are involved in this sort of packaging and degradation pathways?
It's really any type of receptor. It's pretty generalized to just all of them. So when they're not needed, they can just be sorted inside the MPV and then sent wherever they're needed.
So you had your future directions.
Yeah.
But there was the one slide where you were like, we got this result and we didn't know quite what it meant and we ran out of time and all that. Can you go back to that slide? And just assume, so it's the one right in the middle with a plus wild.
Plus wild.
So let's just assume that was, you did that like 20 more times and it looked exactly the same.
And these were all of triplicates.
Okay, and so what do you think is happening there?
So we don't think that the cells were taking up the adding. So it was in a strain of yeast that was missing VPS4. And when we tried to add VPS4 back in, we wanted to see how well it was taking up that wild type VPS4. So we want it to look like this because that's a real wild type cell. But this is showing that our results aren't actually like wild type. We weren't able to get that same complementation of adding the wild type VPS4 back in. So it would be interesting to see if we would have done, a few more replicates to see how it would have turned out. We were starting to see trends that were more similar to wild type because this one almost looks more membrane bound than soluble. And one of the last westerns that we ran, we did see slightly more soluble than the membrane bound, but it wasn't completely night and day like that.
So I sort of have a follow-up on that question, maybe it's a two-question. So what implications does that have for the other methods? Is it done in the same way? So
they are done in the same way. So I know it's kind of like questioning my results that I got with the VPS4 mutants. So we would still need to run more replicates of this as well, because if we're not getting complementation that looks like the actual wild type, I mean, how accurate are these with incorporating those mutants in? But when we saw, and I guess that goes back to the microscopy pictures as well, because we're seeing that.
That would be the next question I have. So is this wild type a real wild type, or is it, again, did you do any microscopy on a one where you tried to replace the wild type?
I think we did, and I think it looked more like our mutant forms as well. So it wasn't looking at just like the wild type version.
So both might be indicating that you have actually an experimental problem rather than.
Yes. And I think we're really trying to be thorough with how we're running each of these and being really accurate, so we're staying consistent.
There's lots of good information besides that, so I didn't want to imply otherwise.
How do you go about getting GFP tagged onto that CPS receptor?
I actually didn't do that.
Oh, okay.
We had it, we probably bought it that way, so we were just able to use it. CPS is a really common, it's more of a cargo that can be sorted by receptors. So we're just able to see where that cargo is going. Yes?
Back to the picture with the that part. So the polymer part, is that the S43? Is up the polymer?
Yes, so S43 is the purple spiral, and then over here it's just broken into all of its pieces.
Does it have the same monomer units all the time? Is that what defines it, or is that variable?
It has the same, so this is a simplified version of it. So it has those 7 subunits, and then there are multiple copies of each subunit. So it does have all those parts consistent.
So it's consistently the same thing. It's not, there's not much variability at all.
Correct.
With that polymer. Okay. That's a question.
And they're still working on, specific shapes of this as well. There's not much on the definite structures of BPS4 or BTA1.
And that would go back to why you want to do the
X-ray crystallography. Yeah, they know that the BTA1 forms a homodimer with itself, and that's what allows the BPS4 to bind to it. But they're still working on structures. Other questions?